Hi, thank you for joining. We will wait a few minutes for people to connect. Good afternoon and welcome to NRGene's webinar series. My name is Yael. I am part of the marketing team and I'll be your host for today. In today's webinar, we'll learn about the challenges in the maize breeding program in Brazil and how NRGene solutions can help solve those challenges in the breeding world. We will also present interesting results from a recent project. We will save time for questions at the end of the webinar, so we encourage you to type in your questions during the presentation using the Questions tab on the left of your screen. This webinar is being recorded and will be posted on our website in case you want to listen to it again. It's my pleasure to present today's speakers. Our first speaker is Dr. Ivan Shuster. Ivan is an agronomist and holds a PhD in genetics and plant breeding with 20 years of experience in the seed industry, mainly in molecular breeding. During this time, he served as an R&D leader at Coodetech, Dow AgroScience, and Longping High Tech. Currently, Ivan is the leader of molecular research for corn breeding at Longping High Tech, Brazil. I would also like to welcome Dr. Kobi Baruch. Kobi has been with NRGene since 2011. In his current role, Kobi leads a team of scientists and bioinformaticians and oversees all genomics analytical projects in the company. Kobi holds a PhD in bioinformatics and molecular genetics from the Hebrew University and has extensive experience in computational and molecular biology.
Hey, good afternoon. Thank you for the introduction. I want to share some of our experience in plant breeding and the new technologies for plant breeding, especially for molecular breeding and the opportunities and the challenges that we can see when we use molecular breeding. Our experience during the last years, I think, is very useful in introducing molecular breeding in corn breeding, plant breeding, as a tool to accelerate the genetic gain. And to show how it is possible to accelerate the genetic gain in the plant breeding using molecular breeding, I want to share some of the opportunities that molecular breeding have in corn breeding. Basically, I used to divide the plant breeding in 4 stages. That is the first one that we call the discovery. That is the stage where we created the variability in the germplasm. And after creating the variability, we pass for the second stage when we select the better recombinants. This 2 first stage is very close to R&D, specific to R&D and specifically for breeders or for the teams that is related to breeders. And the other 2 stages is for testing when we involve some other groups for testing the better products that was selected in the first stage in a huge number of locations to look for the adaptability of the new products. And it is also making between R&D and marketing, that is normally a product development stage. And the last one is the launch, that is more related to marketing and commercial. This is the 4th stage that plant breeding can be explained in a seed industry, from creating the variability until to reach the market. Each of these stage can take some years, but basically we can summarize the plant breeding in this 4th stage. And as an example for corn, when we are creating variability, it is the way that we use to create the breeding populations. And then breeding populations are being created by crossing some parental lines or making some different kinds of populations by mixing or by crossing or something like that. And then creating a segregating population. This can be self-fed until to reach a high level of homozygosity, or also advance the homozygosity by double haploid products. And then we pass for the second stage, that is to select the better hybrid line that was created in the discovery stage. And then in corn, normally the hybrid lines are being selected by crossing with some elite testers in a stage that is we call top cross. And then we evaluate all of the hybrids between the new hybrid lines with the highly elite testers, and selecting the better hybrids. We are selecting the better new hybrid lines. Then the top cross, the main objective is also to identify some very good hybrids to advance for the next generation. But the most important information that we have in the top cross is the performance of the new hybrid lines. And then when selected the better hybrid lines in this stage, we can create new hybrids with these hybrid lines for the testing stage. Then testing stage will use the better hybrids that can be advanced for this stage of top cross, but also new hybrids that will be created based on the information generated in this testing stage of the new hybrid lines. And then after this testing, that can take some years in different environments, the better hybrids will be selected to go to the market and then to be launched. And in a very simple way, we can simplify plant breeding in this stage. And how each stage is important and how we do in each stage. In the first stage, when we are looking for discovery, normally in the breeding program, especially for corn, the information that we use to create the better population, that could be population derived from crosses or population derived from mixing some parental lines like the synthetic population. The information that the breeders have is the pedigree of the parental lines, also the combining ability for the parental lines. Then they know how parental lines can combine in average with lots of other parental lines. Also, the enterotic groups of each parental lines for breeding population is known. Also, information from some progenies. If the breeders knows that some of the parental lines already produced some good progenies, it could also be used as a parental lines for new breeding population because his performance was already tested as a good parent for breeding population. And also have some information for specific traits like disease resistance or the earliness of the harvesting time. This is normally the information that the breeder have to create their breeding population. Using molecular breeding, we also can provide other kinds of information that can help the breeders to be more precise to select the parental lines for the breeding population. Based on the genotype of the parental lines, it is possible to simulate a breeding population based on the genotype of parental lines in a two-way, three-way, or four-way, or some design of crosses. We can simulate the genotype of the double haploid lines or the highly homozygous lines after some selfing generation. But normally, based on double haploid approach, and then we can identify the variability or the genotypes, or can simulate the genotype of the double haploid lines, and then we can evaluate the variability, the average, and the variability that we can obtain in different breeding population based on the divergence between the parental lines, and based on the genotypes of the parental lines, we can simulate the genotype of the progenies, and then make predictions about the performance of the progenies. And then look, this blue line is predictions of the performance of one breeding population, and the orange line is the predictions of the performance of second breeding population. Then breeders can, when comparing these two possible breeding population, they can select how is the better breeding population that have a higher average. Not only a higher variability in the breeding population is good as they desired. Also, in this case, we can have less variability, but with a higher average. Then breeders can focus on this population and just drop this population before making the crosses, before creating the breeding population. Then, based on this approach, we can create a huge number of breeding population to evaluate it in silico, and then just to select the better predicted breeding population, and then make the crosses and create the population to evaluate it in the field. Then it's possible to evaluate much more possibility of breeding population than we used to do in the current approach using just the informations before having molecular data. When we move for the second stage, that is the selection stage, represented in corn by the test cross or the top cross. we will cross all of the new hybrid lines with some elite testers and then evaluate it in the field, in the yield trials, and select the better hybrids and also the hybrid lines that have the better combination with the elite testers. What is the information that the breeders have to create his top cross? They knows very well their testers, that is some elite lines from these breeding programs, and they also knows the heterotic groups of their testers. And based on some previous information, especially from the pedigree of the hybrid lines, they can also estimate the heterotic group of the hybrid lines. That is the information they have to define how they will proceed with the top cross. When we add some molecular information at this stage, it is possible to simulate or to predict millions of hybrids before making any cross between hybrid lines and top crosses. Just simulating the cross and then looking for the genotype that is expected for the hybrids, because we need to have the genotype for the parental lines, and having a good prediction model, we can predict the performance of millions of hybrids at this stage. And with this predicted information, breeders can select the better hybrids that he can have before create the hybrids to be evaluated in the field. Then we call this the first generation of selection, also is made in silico trials. We can create millions of trials in silico that we cannot create in the field. And then we can have a huge number of hybrids to be evaluated in the first generation. As the accuracy of this approach increase, we can just skip the top cross generation and pass directly to the testing stage. Then we can accelerate the breeding program. Also, when we are making millions of predictions with very new hybrid lines, we can identify some good candidates for testers, for new elite testers, based on the GCA for the predicted hybrids. And it is a way that we can have an evaluation. This is some examples of some thousands of hundreds of hybrids predicted, and how we can select just the better performance in silico, and then move to the field to evaluate and then select the better hybrids also in the field. But we can go to the field with very better hybrids. Then the average of the yield trials in the field will be higher, because we already dropped the hybrids that will have a bad performance in the field. Sorry, a worse performance in the field. In the same stage of testing, the second stage of what I call in the breeding program is to define the heterotic group of the new hybrid lines. Breeders normally do it based on the pedigree, the parental lines, the heterotic group of the parental lines. They can assume the heterotic group of the hybrid lines, and also they will adjust this classification after some field evaluation based on the GCA, or the ability combining of each hybrid lines when crossing with different testers for different heterotic groups. Using molecular markers, we can identify these heterotic groups very fast, very early in the breeding program, before having enough seeds to make crosses. We already can classify the new hybrid lines in the heterotic group based on the molecular information, and also based on the knowledge of the germplasm of the heterotic groups we have. And sorry, and this classification is very accurate when you use a molecular marker to classify the new hybrid lines in these heterotic groups. When moving for the next stage of breeding program, that is for testing, what currently in the breeding programs for corn is used is the better hybrids from the top cross will be advanced for a new round of testing in more locations, and also with more reps sometimes. And also some of the better hybrid lines selected in the top cross will also be recombined or be crossed with other testers to create new hybrids to be tested in this stage. And what the breeders have the information to move to the testing is the field evaluation of the top cross, and then the knowledge of the performance of the hybrid lines, and also they will consider to create the new hybrid lines, the GCA for the hybrid lines, and also the heterotic group of each hybrid lines. When using molecular data to help in this stage, we can make better prediction for this testing of generation because we can use the data from the first trials for the new hybrid lines, and then using this field information, we can improve the prediction models for these specific hybrid lines, and then having a higher accuracy to select the hybrids to be tested in the next generation in more locations and with more reps. It is because as close as the hybrid lines from the training population related to the population we are predicting, better is the accuracy of the model. And when we use, for example, this top cross yield trials as a training population for the hybrid lines that was being involved in this training population, we can use a new model with this training population to predict better hybrids with these new hybrid lines with other testers, and also between new by new to create better predictions for this testing stage. Then we can have much better hybrids also in this testing stage. And also using this new model, using this training population from top crosses, we can identify some very better or some very good testers in the new hybrid lines just created. How this molecular information or how molecular breeding can impact in the genetic game. This is the breeder equation. All of the breeders know how it works. Then the genetic gain can be expressed as the intensity of selection, the accuracy that is translated as the root square of the heritability of the trait, and the additive deviation that is also square root of the additive variance. And in the denominator, we have the number of years for one breeding generation from starting the process for breeding population until advance the hybrid to the last testing stage. And then when we use genomic prediction, we can predict a huge number of hybrids, and then we can select. It can be very restricted in the selection. Then we will increase the selection intensity many times, and increasing intensity, genetic gain will be increased. Also, when we are making prediction using molecular markers, the molecular markers are not affected by the environment. Then also the heritability of the predictions is high, and then when we increase the accuracy of the predictions, also the genetic gain increase. Also, when we are managing a huge number of hybrid lines and hybrids with molecular markers, we can quantify the variability in the population, and also we can manage the genetic variance, or at least the additive variance, and also we can increase this part of the equation. And using prediction, we can accelerate, we can skip some steps on breeding and reduce the number of generations. Then all of these improvements is directly related to genetic gain. Then molecular breeding have lots of ways to increase genetic gain in the plant breeding. What is the state of the art for molecular breeding? Currently, the main breeding companies use molecular breeding in their programs worldwide. The methods that they use is currently very well known. Each company can have their own design, but the overall method for molecular breeding is known and very well published in the literature. For sure, that is, I can say it's not new, but it's pretty new. And then improvements is being made continuously, and the industry also recognize the advantage of molecular breeding for the seed industry. The challenge for molecular breeding especially is related to applying this technology in the large scale. What is some of our needs? We need to have a very good phenotypic data for training population to create a high accuracy model. We need to have a good approach to account for genetic bioenvironment interaction to not create some bias in our predictions. Also need to have a very good genotyping platform with very good genotypic data to produce the huge number of molecular data that is needed for molecular breeding. And also it is necessary to have a very good data managing system because the number of data that is produced is really very big. And one of the big challenge is the cost for genotyping, because we know genotyping cost is being reduced, but also the genotype data that we need is very high. When we are looking for the one free statement that I created for genomic selection, I can say, if you have a very good coverage of the genome by molecular markers, it will be at least one molecular marker in leakage equilibrium with each QTL in the genome. And then if you have this situation, the prediction model will capture theoretically all of the QTLs for the specific trait. Then the accuracy of the model will be high. But to have this high coverage of the genome, we need to have a huge number of markers. Sometimes we already have some experience testing the number of markers for models. It needs to be at least 5,000 markers. Sometimes models use more than 5,000, but at least 5,000 markers in corn that have less than 2,000 centimorgans in their genome. It's can say a good number for having a good predictions. As I said, genotyping cost is reducing very significantly in the last years. We can have currently one data point costing just a fraction of 1 cent, but also we need to take into account the cost of samples, because the cost for data point, it no makes sense when you are looking for a huge number of markers. Then at the end, what matters really is the cost per sample. If you have a very cheap value for data point, but we need to have a huge number of markers and a huge number of samples, the cost per sample will still be high. For sure, the molecular breeding benefits is high, and even having a high cost for genotyping, the benefits outweigh the cost. But when we are starting a molecular breeding program, we need to know that the financial benefits is not immediate. If we have some genetic gain increase using molecular breeding, it will be observed in the launching of new hybrids some years later, and also the market benefits will be also observed some years later after launching. Then we are looking for an increase of the cost at the beginning, with the benefits with some years in front. It is still an advantage to use molecular breeding, considering the cost, but in all of our professional activities, we are all the time looking for increasing our efficiency and increase the resources that we use, increase the efficiency that we use these resources. And this is an opportunity for molecular breeding when we look for imputation. We started last year in a pilot project with NRGene in the imputation to understand how imputation could help molecular breeding. Our goal when we are looking for imputation is to reduce the number of markers in our genotyping panel. If we can reduce significantly the number of markers in our panel, we also can reduce significantly the cost for genotyping. That is the main target. What we want to know is the imputation really works. The efficacy of the imputation is really high. We can have a good confidence in the data for imputation. And that was we did in 2020 with NRGene and our first pilot for genome imputation. And the results is very, very good. We have a very high accuracy in imputation the genotype comparing the genotype in the same samples. And what I mean is genomic imputation will be a new routine in the molecular breeding programs. It is a way to have a more efficient use of the R&D resources to accelerate the genetic gain using molecular breeding, and the cost for molecular breeding cannot be so high, and the breeding programs can use the resource to increase also other areas that need to be accelerated or need to be improved in the R&D status. That is what I want to share with you in this time, based on the experience of molecular breeding we had in the last years. And then I will ask to Kobi to follow with his presentation about genomic imputation. And we will have a Q&A session after Kobi's presentation. Thank you.
Thank you very much, Ivan, for a very, very interesting talk. So here in NRGene, we've followed a lot of breeding programs for many crops, and we've noticed that they all share similar challenges as you presented right now. We recognize these challenges and developed dedicated solutions to mitigate them. In this part of the webinar, I'll present results from recently delivered projects and demonstrate how we have solved these challenges. I'll explain how our solution not only reduce costs, but also potentially enhance the quality of genotyping, and of course for downstream applications. Okay, so just a brief overview of NRGene, the company. So NRGene is an agtech startup founded in 2010. Our first line of products and services were launched to the market at 2015. We have a vast variety of projects coming from 138 customers in over 30 countries. And we've worked on 79 species up until now. And some of those projects were also published in peer-reviewed publications, which are, of course, available to read. And we have links in our sites for your convenience. So a bit about our product line. NRGene sees as one of our main objectives to provide a holistic solution for molecular breeding. More specifically, we wish to lay out all the infrastructures which are necessary to conduct a very highly efficient and accurate molecular breeding. And to do so, we provide a range of products and services from the very fundamental reference genome assemblies through Pan Magic, which is a product for comparative genomics of several genomes. From there, moving to diversity analysis and investigate the genetics of a whole germplasm of a breeding entity. And next, we also provide Snipper, which is a genotyping, high throughput genotyping solution. And once we have this capability, we can move onwards to predictive genomics, which is represented here by the product Trait Magic. And finally, we also provide decision supporting platforms for breeders in the form of Genome Magic, which is a breeding platform. As we mentioned earlier, the main topic of this conversation is going to concentrate on the Snipper. In the next slide, I'll give a brief description of the product, and later on, as I mentioned, I'll dive into a real use case in a project that we recently done for a customer. So what is Snipper? Snipper is a product aimed to design an optimal customized marker set for a specific breeding entity. It is customized in the sense that we design those markers on the base of the client's germplasm. It is aimed to high throughput genotyping, meaning the process is designed to be optimal in accuracy, cost, and turnaround times to high throughput genotyping. And we are dedicated to make this process as accurate and efficient as possible by creating the customized optimal SNP set for the purpose the customer require. So why use Snipper? Of course, you need high throughput genotyping for all the good reasons Ivan also mentioned in his talk. You want to make rapid advancement in your molecular breeding program. You want to reach maximal accuracy, which will lead later on to improved quality of both your genotyping and the downstream applications which utilizes this genotyping. You would enhance your selection intensity and diversity management. And of course, all of that will eventually lead to reduced operational effort, turnaround times, and direct and indirect costs, which are related with high throughput genotyping. So this is a very abstract the schematic description of the Snipper process. There are, of course, a few parts done by NRGene, which is the actual Snipper, here in the gray shaded rectangle. So first of all, we get from the client his haplotypic collection of germplasm. This collection is then being processed with a diversity analysis to characterize the diversity. This stage is usually done using millions of SNPs. Next, the next module is actually using this diversity panel, this diversity analysis in order to extract an optimized information content for each of the markers, filtering the markers with low information content, and subsequently also designing a SNP set of thousands of SNPs, which we call during this process target set. Another step, which is optional in this stage, is out of the target set that is composed of thousands of SNPs, we can also actually calculate the minimal optimized set of a few hundreds of SNPs for imputation purposes, and this set is termed minimal set in our process. Eventually, after these two stages, which are the design stages, and research stages. We reached the third and final stage of the Snipper, which is the actual routine genotyping, meaning the production level genotyping, which is done in high throughput manner, usually on tens of thousands of samples every year, in a very fast, accurate, and low cost manner. These results from the high throughput genotyping is then fed back to the customer's system, which conducts downstream analysis using this genotyping data, eventually leading to fast new product launches. So as I mentioned just earlier, the diversity analysis is the first part done by NRGene, and as illustrated here in this slide, you can see that we actually analyze all the haplotypes existing in the germplasm, in the input germplasm of the customer, meaning we are designing the markers in the downstream sets to be fully informational for the dedicated germplasm of the customer. Out of that diversity analysis, we come up with both the target set, which is thousands of SNPs, and the minimal set, which is hundreds of SNPs. And then we move from the left side, which is the more research-based stage, to the routine stage, the production level stage, where we genotype the tens of thousands of samples each year with lower resolution, and then impute it back to higher resolution panels and deliver it to the customer for downstream applications. So now that we've went through a brief description of the Snipper product, let's dive into a real use case of a recently completed project for a customer. So this use case was involving maize as the crop of interest. And the objective of this project was to design an optimal minimal set, meaning hundreds of SNPs set from an actual existing, a pre-existing target set, meaning the customer has already provided us with a larger set of thousands of markers he has been using, which were very expensive for genotyping, and NRGene's objective was to extract out of those the minimal optimized set in order to impute back to the target set. The turnaround time had to be very fast, under 4 weeks for this project. The input data for this project was, as I mentioned, the target set of a few thousands of SNPs. These were raw genotyping results that we got. We also got these genotyping results for the target set for roughly 10,000 double haploid lines, which are made from more than 150 cross populations. Those populations were 2-way, 3-way, and 4-way crosses. And then once we got this input, we went into the actual process of the minimal set design. This process was composed first from QA of the input data, followed by filtration stage of the off types or mislabeled progenies. Next, we corrected the parental and the progeny genotyping by comparing them to each other and actually completing missing data points and fixing wrong data points. Next, we ran a proprietary algorithm to design the optimal minimal set. And then after, we of course entered a stage of the actual imputation and assessment of the accuracy of this imputation by comparing the in silico imputation results that we got to the actual genotyping results that we had. So these are some examples for the QC and filtration stages of the progeny. In this graph that is presented here, we see 4 populations out of the 150 populations that participated in this project. And as you can see, it's a PCA analysis. We see here PC1 and PC2 in the X and Y axes respectively. And you can see that these populations are quite spread along the PC1 axis, which might indicate some noise or mislabeling of those samples. And indeed, when we dug into the data, we found out that a lot of those samples are suffering from very high rates of no-call data points. So we filtered those out, and as you can see, there was a dramatic shift in the pattern of the PCA. Again, these are 4 populations, but it was the same picture for all 150 populations in this assay. Next stage was to correct either wrongly genotyped data points or data points with no call due to technical problems during the raw genotyping process. And in that process, approximately 10% of all parental data points had been corrected, where most corrections were indeed replacing no calls to actual genotyping values. And in the case of the progeny, 12% of the data points had been corrected, again, mostly replacing no call to actual genotyping values. This process, of course, is very important because, as we are continuously mentioning throughout this webinar, cost effectiveness of this process is crucial in actual breeding programs, and every data point counts. Eventually, we ended up with a minimal set of around 500 markers. Here we see an illustration how those markers are spread along the chromosomes of the maize. Of course, the main objective here was to genetically spread them evenly on the genome. And eventually, the last stage was to actually conduct the imputation and assess the accuracy of this imputation compared to the real results, real genotyping results we have for those samples. So again, what we do here, we took more than 150 populations with the minimal set, meaning we used different minimal sets. As you see here in the x-axis, we started off with minimal sets size of 20 and went up to 1,000 SNPs in the minimal set. And then we used those sets to impute those 150 progenies back to the target set, which is 5,000 markers, 5,000 SNPs. And on the y-axis, what we see is the actual percentage of correct imputations compared to the real data that we have for genotyping. And of course, as expected, the more SNPs you use in the minimal set, the higher the accuracy. And the main objective of this drill is to find the optimal cost-effective point where you genotype a reduced set of SNPs, but still get high accuracy in that process. And for example, here we can see that with 500 SNPs in the minimal set, we get an average accuracy of 97.6, which is very nice. Next, here we demonstrate that this accuracy is not evenly distributed along the different types of populations. As I mentioned earlier, we had in this project more than 150 populations coming from two-way, three-way, and four-way hybrids. And as you can see here, and as expected, of course, if we are referring here, for example, for the 500 markers in the minimal set benchmark, we see that for the four-way hybrid, we get only 95.7% accuracy, and this number increases as we approach to the two-way hybrid, which is naturally the highest accuracy. So the results we saw in the earlier slide was referring to the average of all three types of populations, but still for all three types, we still get respectively high accuracy using the imputation. So to conclude this part of the talk about this use case that I've presented, we finally chose the minimal set to include 500 SNPs to be imputed to the 5K target set, meaning it's a ratio of 1 to 10. We reduced the actual genotype data points by a tenfold. And the genotyping costs per samples in the production stage are expected to drop by at least 2 times, meaning half the price, using this approach of minimal set, which is imputed to a target set. So at this slide, I wanted to kind of review with you the related costs to Snipper and the different components of this product. So when we're talking about target sets in different breeding programs of different crops, it's usually a set of thousands of SNPs. And those usually cost in practice per sample tens of dollars. The most common molecular method used to genotype these scales is hybridization arrays. The second component, which is a minimal set, which is hundreds of SNPs, usually costs a few dollars per sample, mainly using amplicon sequencing methodologies. And then we have also the computational part of the imputation, which is also a few dollars per sample or even less, and that price is also depending on the number of analyzed samples, of course. So the take-home message from this slide is actually lower density genotyping combined with imputation can actually significantly reduce costs compared to alternatives of higher density direct genotyping. So to summarize my talk, I would like to review again the advantages of the Snipper product. So as I mentioned, you get more accurate data by using Snipper. The genotyping is optimized to the actual germplasm of the breeding program, which means that the data points that are used in the set are much more relevant and informative regarding the breeding materials that you're using. We also, of course, remove and fix the genotyping data as part of the service. It is custom and flexible, meaning as I showed, the Snipper is composed of 3 main parts: the diversity analysis, the design of the SNP sets, and eventually also the production level genotyping. And we customize those 3 stages all together or separately for the needs of the customer. The cost reduction, of course, is a function of all of the above, and we aim eventually to give better genotyping results for less money, which enables the customers to do more for less. And the ease of use of the Snipper is actually by getting to customers an all-inclusive solution, one stop-shop solution, which is also cost-effective genotyping for their needs. And with that, I would like to conclude and thank you all for attending this webinar.
Thank you, Kobi, for that presentation. We have some time now to answer some questions from the audience. The first question, this is for you, Kobi. Will the marker set still work if I acquire new lines or germplasm?
Thank you, Yael, for this question. Very good question. So basically, the answer is yes. But it also depends on the actual diversity of the new lines that you introduce into the germplasm and how diverse are they compared to the germplasm that was used to design the target set and the minimal set. The snipper is flexible in the sense that you can also alternate the target set or the minimal set in case you want to introduce new diversity into your germplasm. So the answer is yes.
Thank you. The next question is directed to Ivan. How are you predicting specific combining ability, heterosis, dominance with molecular markers in silico populations?
There is some modules that we can use to predict additive plus dominant models, and it is kind of a routine that we can use. In this case, when predicting the performance, yield, and other traits for the hybrids, and giving back the average of the yield of each hybrid from each single hybrid lines, we can calculate the GCA based on that. But the model used to predict hybrids is not just additive or not just dominant, it's an additive dominant model. And this gives us the data that we need for GCA calculation. It is the same way that we can calculate GCA from the field data, just replacing the field data from the predicted data.
The next question is again for Kobi. How are you computing imputation accuracy? Oh, I'm going to repeat the question. How are you computing imputation accuracy?
Okay. So the stage of assessing the accuracy was enabled in this case because we actually had the real genotyping information coming from the target set of the progeny samples as well. So we had the real solution, which is the actual genotyping results, and we used the minimal set to impute back to the target set and compared the in silico results of the genotyping to the actual results coming from the genotyping, the full genotyping of the target set of those same samples.
Is the minimal set including imputation during the selection, or all of this is imputated? Sorry, once more, is the minimal set including imputation during the selection, or all of this is imputed? We're having some technical difficulties, but we will get to all your questions in a follow-up email after the webinar. I'd like to again thank Yvonne and Kobi for their time today. Thank you for sharing with us your presentations. This webinar was recorded and we'll have it available in the next few days, so keep an eye out for our email with a link to the recording. It will also be up on our website as well. If you have further questions or an interesting research project you'd like to discuss with our experts, please don't hesitate to contact us at nrgene.com. Thank you for joining us today and enjoy the rest of your day.